Hello again, Jared Pitts here, and today I'm reading another article by Dr. Peat. This one is on his website in the article section, raypeat.com. And the title of this is quite long. Here it goes. BSE, mad cow, Scrapie, etc. Stimulated amyloid degeneration and the toxic fats. So, just to break that down, BSE, that's bovine, bovine spongiform encephalopathy. I think I said that correctly. Mad cow, and I think BSE and mad cow are kind of interchangeable, if I'm not mistaken. And then Scrapie, I had to look up, and this is a fatal degenerative brain disease in sheep and goats. And it's also caused by proteins that are folding incorrectly. I believe similar to mad cow. Anyway, he's going to get into this in this article. I've been extremely interested in this topic for the past couple of months, maybe not that long even, but very recently, and especially because of the neurodegenerative diseases in humans, things like Parkinson's, Alzheimer's, ALS, Huntington's, things like this. And so I've been kind of doing a deep dive into carbon monoxide, not dioxide, monoxide, and the effects, the genetics, the quote-unquote genetics behind this, and what could cause someone to express this protein folding. So, man, I want to get to the bottom of this because this is some very interesting stuff. And I think Dr. Peat maybe understood this better than 99.9% of people. So this is how it goes. I have written before about the protective effects of carbon dioxide and progesterone, especially for the brain, and how the structure of cell water is affected by adsorbed and dissolved materials and by metabolic energy. In the high energy rested state, cell water behaves as if it were colder than its real temperature, and this affects the behavior of proteins and fats in the cell, allowing quote-unquote oily surfaces to remain in contact with the more orderly water. Carbon dioxide spontaneously combines with the amino groups in proteins, stabilizing the normal functional conformation. The loss of carbon dioxide affects the structure of all proteins in the body, and the loss of cellular energy affects the structure of the intracellular proteins and their associated molecules. In Scrapie and many other degenerative diseases, the amyloidoses, proteins condense into fibrils that tend to keep enlarging, with a variety of very harmful effects. The condensation of the amyloid proteins is sensitive to temperature, and a slight increase in the disorder of the water can induce functional proteins to change their conformation so that they spontaneously associate into fibrous masses. In the absence of sufficient carbon dioxide, all proteins are susceptible to structural alteration by the addition of sugars and fats and aldehydes, especially under conditions that favor lipid peroxidations. The amyloidoses affect different tissues in different ways, but when they occur in the brain, they produce progressive loss of function, with the type of protein forming the fibrils determining the nature of the functional loss. The protein which carries thyroid hormone and vitamin A, transthyretin, can produce nerve and brain amyloid disease, but it can also protect against other amyloid brain diseases. In Alzheimer's disease, Parkinson's disease, Huntington's disease, and the quote-unquote prion diseases, Scrapie, Kuru, CJD, BSE, etc., amyloid particles are formed by different proteins. The transthyretin protein, which is binding small molecules, resists condensation into the amyloid fibrils, but without its normal vitamin A and thyroid hormone, it can create toxic fibrils. And that's from Ragu et al. 2002. Around 1970, I read E.J. Field's suggestion that aging tissues and tissues affected by viral diseases showed some similar structures. quote-unquote inclusion bodies under the electron microscope. In following up those observations, it turned out that old tissues appeared to develop antigens, identical with or similar to Scrapie infected young tissues. The premature aging caused by removal of the thymus gland in newborn animals produced similar results. Field's group and others, e.g. Alpers, were clearly showing that the Scrapie infection involved proteins but not viruses with nucleic acids. In one of the Field's last publications, 1978, he even suggested that the infectious process might depend on a structural rearrangement of the host's molecules, similar to the idea which is now known as the prion hypothesis. Field's suggestions was an important advance in the theory of aging, and the evidence supporting it is now voluminous, but that work has been omitted from the official histories. Although phenomena of quote-unquote imprinting and non-genetic inheritance had been established earlier, the dogmatism of genetics led the scientific establishment to reject everything that challenged the primacy of DNA. When I mentioned to my professors in 1971 the evidence that Scrapie was transmitted without nucleic acid, I could see from their reactions that it would be a very long time before much progress would be made in understanding the degenerative brain diseases. When the exact structure of the quote-unquote infectious protein was later worked out and the 1997 Nobel Prize awarded to Stanley Prusiner, I was surprised that no one from Field's group was included. In 1976, a Nobel Prize had been awarded to D.C. Gajusik for his promotion of the idea of quote-unquote slow viruses in general, and particularly for arguing that Scrapie, CJD, and Kuru were caused by slow viruses. In reading Prusiner's autobiographical statements, I was even more surprised to see that he claimed to have been puzzled to find out, around 1983, that the infectious agent was a protein. I had thought that my professors were lethargic authoritarians when they refused to look at the evidence in 1970 to 1972. But Prusiner's expression of puzzlement so many years later over the absence of nucleic acid in the infectious agent is hard to account for. In my own research in 1971, I was interested in another kind of age-related quote-unquote inclusion body, which was variously called lipofuscin, age pigment, and ceroid pigment. This brown, yellow autofluorescent pigment contained proteins and metals, as well as polyunsaturated lipids, and overlapped in many ways with the amyloid bodies. All of these inclusion bodies were known to be associated with radiation injury, aging, and hormonal nutritional imbalances. Excess of estrogen, polyunsaturated fatty acids, and oxidative metals were major factors in the development of lipofuscin, and estrogen was also known to cause other types of quote-unquote inclusion bodies to develop in cells. Although very little was known about the composition of the inclusion bodies, they were usually thought to be organelles damaged by free radical activity or antibodies resulting from autoimmunity. Their involvement in aging and degenerative disease was clear, and it was widely known that ionizing radiation accelerated their formation. But it was just at this time that the national research priorities of the U.S. were redirected toward genetic explanations for all major diseases, with, for example, the quote-unquote war on cancer, centering on the concepts of the quote-unquote oncogene and the cancer virus. Since the quote-unquote slow virus of cancer, or the viral oncogene, requires activation by something in the environment, its function is to distract the public's attention from those environmental causes of disease, viz., radiation and chemical pollution. The U.S. Public Health Service has historically been one of the branches of the military and currently has 6,000 commissioned officers. It has been intimately involved in all aspects of chemical, biological, and nuclear warfare, and it has participated in many covert projects, including experimentation on people without their knowledge. For decades, information on radiation injury to the public was hidden, classified, altered, or destroyed by the PHS. During the radiation disaster at 3 Mile Island, they calmly defended the interests of the nuclear industry. After the April 1986 catastrophe at the reactor in Chernobyl, some of the food being imported into the U.S. was so highly radioactive that the FDA secretly seized it to prevent the public from being concerned. The first cow found to have BSE in England was in November 1986, several months after England's pastures had been heavily contaminated by rainfall carrying radioactive materials from Chernobyl, which soaked into the soil and continued to contaminate crops for years, and will continue for centuries. The number of sick cows increased rapidly to a peak in 1992. Human deaths from the similar disease, variant CJD, began a few years later. In June 2000, a wildfire burned across southern Washington, turning the radioactive vegetation on the Hanford nuclear site into radioactive smoke, contaminating a wide area, including farms, dairies, and orchards. In 2003, the first cow in the U.S. with BSE was reported from a dairy a few miles from the Hanford site. Beginning in 1946, Bikini Island was used to test atomic bombs. In 1954, they began to test hydrogen bombs in the Pacific. Some of the bombs were deliberately designed to vaporize whole islands, so that the effects of radioactive fallout could be studied. In 1954, the first child with Kuru was reported in the rainy highlands of New Guinea. Within 2 years, hundreds of people in that area, of the Four tribe, were dying from Kuru, with the mortality highest among the women. In some villages, the majority of the women died from the disease. But by 1957, the mortality was falling rapidly. Between 1957 and 1964, 5% of the population of the Four tribe died of the disease, according to D.C. Gaujsik, who had been sent by the U.S. Army to investigate the disease. Although Gaujsik graduated in 1946 from Harvard Medical School as a pediatrician, in his autobiography he said that when he was drafted in 1951, the Army assigned him to work in virology. In 1958, Gaujsik became director of the NIH Laboratories for Neurological and Virological Research. This was a remarkable achievement for someone who had supposedly only done some scattered fieldwork in infectious diseases, and whose purpose in going to New Guinea had been to study, quote, child growth and development in primitive cultures, end quote. The only published reason I have found that might be a basis for making him head of neurology was his sending a diseased forebrain to Fort Detrick in 1957. Oh boy, Fort Detrick. Wow, that place comes up everywhere. Gaujsik claimed to have seen the 4 people eating dead relatives, but his figures show that the disease was already in rapid decline when he arrived. He took photographs which were widely published in the U.S., supposedly showing cannibalism. But 30 years later, he said the photographs showed people eating pork, and that he had seen no cannibalism. At the time Gaujsik was observing Kuru in New Guinea, the influence of quote-unquote cannibalism on brain functions was already in the news because of the discovery of J.V. McConnell that the behavior of quote-unquote trained flatworms could be transmitted to other worms by chopping them up and feeding them to the naive worms. Harvard Medical School, in association with the military program centered at Fort Detrick, Fredericksburg, Maryland, was active in biological warfare in the 1940s. I think it's more plausible to see Gaujsik as a troubleshooter for the biological warfare establishment than as a biological researcher. One of his biographers has written that the idea of associating Kuru with Scrapie was suggested to him by a veterinarian, and that Gaujsik had responded by claiming to have experiments in progress to test that theory, 4 years before the experiments were actually made. In other words, the slow virus theory for which Gaujsik was given the Nobel Prize is scientific junk, which Szent-Györgyi has repeatedly reinterpreted retrospectively, making it seem to have been anticipatory of the prion theory. Whatever actually caused Kuru, I think the Army was afraid that it was the result of radioactive fallout from one of its bomb tests, and that Szent-Györgyi's job was to explain it away. I suspect that Kuru was the result of an unusual combination of malnutrition, the women were vegetarian, and radiation. In the very short time that Szent-Györgyi spent in New Guinea, he claimed to have done studies to eliminate all of the alternative causes, nutritional, toxic, anthropological, bacterial causes, studies that would normally have required several years of well-organized work. I don't think he mentioned the possibility of radiation poisoning. In 1998, Congress commissioned a study of the health effects of radiation from bomb testing, and although the study examined the effects of only part of the bomb tests, it concluded that they had killed 15,000 Americans. No one has tried to accurately estimate the numbers killed in other countries. Even very low doses of ionizing radiation create an inflammatory reaction. That's from Vickers et al. 1991. And there is evidence that the inflammatory state can persist as long as the individual lives. In Japan, the quote-unquote acute phase proteins are still elevated in the people who were exposed to radiation from the atomic bombs. The acute phase proteins that are increased by malnutrition and radiation increase the tendency to form amyloid deposits. Strong radiation can even cause, after a delay of more than a year, the development of vacuoles, which are the most obvious feature of the quote-unquote prion brain diseases. The persistent inflammatory reaction eventually produces cellular changes, but these were originally overlooked because of the theory that radiation is harmful only when it produces immediate changes in the DNA. Radiation damage to the brain is most visible early in life and in old age. In 1955, Alice Stewart showed that prenatal X-rays increase the incidence of brain cancer, leukemia, and other cancers. In 1967, a study in Japanese bomb survivors found that prenatal exposure to radiation had reduced their head size and brain size. In 1979, Sternglass and Bell showed extremely close correspondence between scores on the SAT and prenatal exposure to radiation. Serum amyloid A, which can increase 1,000-fold under the influence of pro-inflammatory cytokines, resulting from irradiation, stress, trauma, or infection, is an activator of phospholipase, A2, or PLA2, which releases fatty acids. Some of the neurodegenerative states, including amyloid prion diseases, involve activated PLA2, as well as increases in the toxic breakdown products of the polyunsaturated fatty acids, such as 4-hydroxynonenal. The quantity of PUFA in the tissues strongly determines the susceptibility of the tissue to injury by radiation and other stresses. But a diet rich in PUFA will produce brain damage even without exceptional stressors, when there aren't enough antioxidants such as vitamin E and selenium in the diet. Amyloidosis has traditionally been thought of as a condition involving deposits mainly in blood vessels, kidneys, joints, and skin, and in extracellular spaces in the brain, and the fact that the quote-unquote amyloid stained in a certain way led to the idea that it was a single protein. But as more proteins, currently about 20, were identified in amyloid deposits, it was gradually realized that the deposits can be identified inside cells of many different tissues, before the larger, very visible, extracellular deposits are formed. There is evidence of a steady increase in the death rate from amyloidosis. It kills women at a younger age than men, often at the age of 50 or 60. Serum amyloid P is called the female protein in hamsters because of its association with estrogen. Castrated or estrogen-treated males also produce large amounts of it, and its excess is associated with the deposition of amyloid. Cohen-Ross, 1985. It can bind other amyloid proteins together, accelerating the formation of fibrils, but this function is probably just a variation of a normal function in immunity, tissue repair, and development. Estrogen increases the inflammation-associated substances such as IL-6, C-reactive protein, and amyloid, and liberates fatty acids, especially the unstable polyunsaturated fatty acids. It also increases fibrinogen and decreases albumin, increasing the leakiness of capillaries. The decrease of albumin increases the concentration of free fatty acids and tryptophan, which would normally be bound to albumin. In the US and Europe, livestock are fed large amounts of high-protein feeds, and currently these typically contain fish meal and soybeans. The estrogenic materials in soybeans increase the animal's tendency toward inflammation, with increased serum amyloid. Officially, BSE appeared because cows were fed slaughterhouse waste containing tissues of sheep that had died of Scrapie. Scrapie was a nerve disease of sheep, first reported in Iceland in the 18th century. When I was studying the digestive system and nutrition of horses, I learned that it was common for horses in Norway to be fed dried fish during the winter. This abundant food was probably used for sheep as well as for horses. The extra protein provided by fish meal is still important for sheep in areas where pastures are limited, but it has now become common to use it to increase productivity and growth throughout the lamb, beef, and dairy industries, as well as in most lab chows fed to experimental animals, such as the hamsters used for testing the infectivity of the diseased tissues. Increased dietary polyunsaturated fatty acids, or PUFA, suppress the activity of the ruminal bacteria, which are responsible for the hydrogenation detoxification of PUFA in the animal's diet. This allows the unstable fats, 98% of which are normally destroyed, to pass into the animal's tissue and milk. The polyunsaturated fats in fish are very unstable, and when they get past the bacterial saturases, or biohydrogenases, in the rumen that normally protect ruminants from lipid peroxidation, they are likely to cause their toxic effects more quickly than in humans, whose antioxidant systems are highly developed. The toxic effects of polyunsaturated fats involve altered immunogenic protein structure, decreased energy metabolism, and many inflammatory effects produced by the prostaglandin-like substances. Marine fish are now so generally polluted with dioxin that in Japan there is a clear association between the amount of fish in a person's diet, their body content of EPA and DHA, and the amount of dioxin in their body. Radiation and many kinds of poisoning cause early peroxidation of those highly unsaturated fats, and the breakdown products accelerate the changes in the folding and chelating behavior of proteins. The accumulation of altered proteins is associated with the degenerative diseases. The role of toxic metals in brain inflammation is well established, e.g. aluminum, lead, mercury, and there's several studies there. The prion hypothesis, and that's in quotes, has the value of weakening the fanaticism of the DNA genetics doctrine, but it has some problems. There are now several examples in which other degenerative diseases have been transmitted by procedures similar to those used to test the Scrapie agent. And there's several studies there. Experimental controls haven't been adequate to distinguish between the pure prion and its associated impurities. Gajusik burned a sample of the infective hamster brain to ash and found that it still retained quote-unquote infectivity. He argued that there was a mineral template that transmitted the toxic conformation to normal proteins. Others have demonstrated that the active structure of the infective agent is maintained by a carbohydrate scaffolding, or that the infectivity is destroyed by the frequency of ultraviolet light that destroys the active lipid of bacterial endotoxin, lipopolysaccharide. But simply injuring the brain or other organ, by injecting anything, will sometimes activate a series of reactions similar to those seen in aging and the amyloidosis. When a slight trauma leads to a prolonged or expanding disturbance of structure and function, the process isn't essentially different from transmitting a condition to another individual. The problem is being quote-unquote transmitted from the initial injury, recruiting new cells and passing the disturbed state on to daughter cells in a disturbed form of regeneration. Keloids, hypertrophic scars, are analogous to the dementias in their overgrowth of connective tissue cells. In the aging or injured brain, the glial cells, mainly astrocytes, proliferate in reparative processes that sometimes become exaggerated and harmful. When tissue phospholipids contain large amounts of polyunsaturated fatty acids, large amounts of prostaglandins are immediately formed by any injury, including low doses of ionizing radiation. The liberated free fatty acids have many other effects, including the formation of highly reactive aldehydes, which modify DNA, proteins, and other cell components. Animals which are quote-unquote deficient in the polyunsaturated fatty acids have a great resistance to a variety of inflammatory challenges. Their tissues appear to be poor allergens or antigens, since they can be easily grafted onto other animals without rejection. Something related to this can probably be seen in the data of human liver transplants. Women's livers are subjected to more lipid peroxidation than men's because of the effects of estrogen, increasing growth hormone and free fatty acids, and selectively mobilizing the polyunsaturated fatty acids and increasing their oxidation. Liver transplants from middle-aged female donors fail much more often, 40 to 45%, than livers from male donors, 22 to 25%, and other organs show the same effect. The autoimmune diseases are several times as common in women as in men, suggesting that some tissues become relatively incompatible with their own body after prolonged exposure to the unstable fatty acids. If we consider the healthy function of the immune system to be the removal or correction of injured tissue, it's reasonable to view the random interactions of oxidized fats with proteins as exactly the sort of thing our immune system takes care of. The serum amyloids A and P and the closely related lipoproteins are considered to be important parts of our quote-unquote innate immunity, operating in a more general way than the familiar system of specific acquired immunities. The amyloids and lipoproteins are powerfully responsive to bacterial endotoxin, LPS, and their structural feature that binds it, the quote-unquote pleated sheet structure, appears to also be what allows the amyloids to form amorphous deposits and fibrils under some circumstances. Our innate immune system is perfectly competent for handling our normal stress-induced exposure to bacterial endotoxin. But as we accumulate the unstable fats, each exposure to endotoxin creates additional inflammatory stress by liberating stored fats. The brain has a very high concentration of complex fats and is highly susceptible to the effects of lipid peroxidative stress, which become progressively worse as the unstable fats accumulate during aging. More than 60 years ago, a vitamin E deficiency was known to cause a brain disease, sometimes associated with sterility and muscular dystrophy. The symptoms of the brain disease were similar to those of quote-unquote mad cow disease, and the condition is now usually called crazy chick disease. Wow, I think I knew a couple of girls in high school that had that. All right, we'll cut that out. Veterinarians are usually taught that it is caused by a selenium deficiency, but it is actually the result of an excess of PUFA in the diet and is exacerbated by increased iron and other oxidants and prevented by increased vitamin E, selenium, or substitution of saturated fats for the unsaturated. Terminology, established by tradition and thoughtless memorization, obscures many of the commonalities in the various brain diseases. Brain inflammation, and we have several studies here, myelination disorders, edema, overgrowth of the astroglia, and circulatory changes are common occurrences in most of the degenerative encephalopathies. But traditional textbook descriptions have created the impression that each disease is pathologically very distinct from the others. The current classification of the quote-unquote prion diseases is reifying a group of symptoms that aren't specific to any specific known cause. And standard laboratory procedures for preparing brain sections for microscopic examination may cause brain cells to shrink to 5% of their original volume. And there's a reference there. So the objectivity of pathological studies shouldn't be overestimated. According to a 1989 study, and it's referenced there, 13% of the people who had died from Alzheimer's disease, and that was in quotes, actually had CJD. Between 1979 and 2000, the number of people dying annually from Alzheimer's disease increased 50-fold. Very competent neuropathologists differ radically in their descriptions of the dementia epidemic. By some tests, the quote-unquote prion resembles the LPS endotoxin. One of the interesting developments of the prion theory is that a particular structure that appears when the prion becomes toxic, the quote-unquote beta-pleated sheet, is also a feature of most of the normal proteins that can form amyloid, and that this structure is directly related to binding and eliminating the bacterial LPS. And if you're not familiar with this yet, LPS, lipopolysaccharides, this is the endotoxin that he keeps referring to. If the prion theory is correct about the conversion of a normal protein into the pleated sheet, it isn't necessarily correct about the incurability of the condition. The innate immune system should be able to inactivate the prion, just as it does the bacterial endotoxin, if we remove the conditions that cause the innate immune reaction to amplify the inflammation beyond control. In the prion diseases, the severely damaged brain appears to have a quote-unquote pathological overactivity of the serotonergic systems. Fraser et al. 2003. This is an interesting parallel to Alzheimer's disease, since it has been known for several years that the blood platelets have an increased tendency to release serotonin in that more common form of dementia. Serotonin itself is toxic to nerves and is part of the adaptive system that gets out of control during prolonged inflammation. Serotonin is an important activator of the phospholipases. The modification of protein's structure by glycosylation is involved in the development of the toxic form of the quote-unquote prionic protein, as well as in all the degenerative processes of aging. Until the ability to use sugar is impaired, cells produce enough carbon dioxide to protect proteins against random glycation. But with each exposure to free polyunsaturated fatty acids, the ability to use glucose is damaged. In the dementias, the brain has a greatly reduced ability to use glucose. One of estrogen's central effects is to shift metabolism away from the oxidation of glucose, decreasing carbon dioxide production. There is a much higher incidence of Alzheimer's disease in women, and estrogen exposure exacerbates all of the changes that lead to it, such as shifts in nerve transmitters, increased vascular leakiness, and the increased production of the acute phase proteins. Everything that is known about the quote-unquote always fatal prionic diseases, the diseases of disturbed protein folding, suggest that they can be avoided and even reversed by systematically reversing the processes that amplify inflammation. People who take aspirin, drink coffee, and use tobacco have a much lower incidence of Alzheimer's disease than people who don't use those things. Caffeine inhibits brain phospholipase, making it neuroprotective in a wide spectrum of conditions. In recent tests, aspirin has been found to prevent the misfolding of the prion protein, and even to reverse the misfolded beta sheet conformation, restoring it to the harmless normal conformation. Nicotine might have a similar effect, preventing deposition of amyloid fibrils and disrupting those already formed. And there's a reference there. Vitamin E, aspirin, progesterone, and nicotine also inhibit phospholipase, which contributes to their anti-inflammatory action, each of the amyloid-forming proteins probably has molecules that interfere with its toxic accumulation. Thyroid hormone, vitamins A and E, niacinamide to inhibit systemic lipolysis, magnesium, calcium, progesterone, sugar, saturated fats, and gelatin all contribute in basic ways to prevention of the inflammatory states that eventually lead to the amyloid diseases. The scarcity of degenerative brain disease in high-altitude populations is consistent with a protective role for carbon dioxide. The relatively sudden acceptability of the idea of non-genetic transmission doesn't mean that Lamarck has been rehabilitated by the scientific establishment. It could just be that it's the most politically acceptable way to explain the outbreaks of deadly disease caused by the industrialization of foods and the exposure of the population to dangerous levels of radiation. And that's the end of the bulk of this article, the remainder being all of those references. So peruse those in your own time, and hopefully you learned something here. This is once again cram-packed. I'll be reading it several more times, and I'm very excited to have found this portion of Dr. Peat's research because, like he said, they want to put a name on everything. And I'm paraphrasing, obviously, but you have all of these different things, all these neurodegenerative, protein-folding diseases, and, well, what if it's all the same source, right? Hope you enjoyed this. Hope you learned something. I'm sure you know someone who could benefit from this. So please share it and like the video here. Subscribe to the channel. And until next time, God bless.